Hi everyone, in this session we are going to discuss some more separation methods like paper chromatography, distillation and fractional distillation. Okay, so in earlier videos we discussed about the separation of water from dye or ink, right? So using evaporation. But is the dye in the black ink or a blue ink a single color? No, usually a dyes or inks are a combination of two or more organic compounds. Okay, so how could you separate it? Using paper chromatography. So what is chromatography? Chromatography is a technique or a method used to separate the colored compounds. The name has given like that because it is initially used for the separation of colored compounds. But after the advancement and technology, there are lot of more advanced techniques of chromatography has been developed because it has immense applications in industries like pharmaceuticals, etc. You will know about chromatography advanced techniques in further classes. But first, you can know in this chapter what is paper chromatography. Okay, for this activity you need a filter paper like this. You cut it this in shape. Okay, so from the bottom edge to up to 3 centimeters we can draw a line with pencil, and at the middle of that line place a ink drop or a dye drop using any capillary tube. Okay, so once you place it, let it dry for some time. Then put this as shown in this picture using a glass rod or any pen. You can use this for the holding. So once it is placed and dipped in a solvent, after development, what happened? Each and every color separated like this. This is the solvent. Water here, the solvent is water, and these are separated spots or the components, and this is what called solvent front. What is solvent front? The solvent carries the different compounds to up front to the solvent flow. Okay, this is called solvent front. Always samples are behind the solvent. Solvent will move fast, then it carries along with this all the samples. Why? How can it carry the samples? Here the simple thing is, whatever the spot you are spotting here is water soluble, is water soluble. Okay, suppose here it is A, it is B, it is C. You can say this is the dye mixture, it is D, you can say. Okay, so why red color moves at the upper front? Why? Because A is more soluble. The component which has more solubility will move faster than remaining. The B has lesser solubility than A. That's why this is the order of separation. The basic point is the component which is more soluble in water moves faster or separate faster than remaining. Okay. Whereas this is the original picture. I am trying to put original pictures from the lab as I have worked a lot of years on this as well in distillation, fractional distillation, so many experiments I have done. So this is the filter paper but spotted not a single spot. Here 4 samples are spotted. After development, they spotted like this. Here in the schematic diagram you have one this thing, no, one spot, no, like that you can place more samples. Where you can apply this? You can separate the colors in dye, pigmentation from the natural colors, even drugs from the blood, right? If you given the blood test, they'll test for the drug test as well, and that component separated like this. But it is a very basic chromatography technique. Usually in academic level, laboratories will run this. Okay. Next method is distillation. What is distillation? Distillation is a separation method used to separate the miscible liquids which has the differences in boiling points. Okay, let us show you with the activity. Okay, distillation of acetone and water. Those are miscible, right? Because water is a polar solvent, acetone is also polar solvent. Both can miscible with each other and boil without decomposite composition. Without any decomposition, they can boil as a mixture. Okay, that's why we have taken acetone and water mixture into a distillation flask, or you can say round bottom flask. Okay, so it look like this. Here in every video I would like to place the original picture from the laboratory. I'm trying hard to find out these pictures because these should resemble same as the activity shown in the textbook, right? So please do subscribe if you find it helpful, right? Then coming back to the point, acetone and water mixture taken in this round bottom flask or distillation flask, and then clamp it like this to the stand. This is the original picture. This is clamped like this to the metal stand. Okay, so at the end or the mouth of this flask, place a thermometer to check the temperature. This is the condenser which has water outlet and water inlet. Why? Because I'll explain. When water is when water income and outcome, this water circulates like this about this tube. Okay, when this circulate, the vapors from this will cool down and condensed. The first one is heating. By taking the mixture in round bottom flask, heat it continuously. Then at what temperature acetone will vaporizes? Acetone boiling point is like 56 degrees centigrade, right? At 56 centigrade, the acetone will vaporize. The vapors come like this, and the vapors go into this condenser. Then because of cooling of this water, the vapors condensed and converted into liquid, and these drops are collected to receiving flask. It can be conical flask or round bottom flask, you can say. Okay, this is also attached to another stand. Okay, this is the setup. So what is the main thing for the separation here? What remains in that flask? Water. Why? Because water boiling point is 100 degrees centigrade. So automatically you will have a doubt up to what temperature, up to what time you can collect this as a acetone, because when temperature rises 56 degrees centigrade, acetone vaporizing start, right? So at that, up to the completion of whole acetone in this mixture, the temperature remains constant at 56 degrees centigrade. So at that time you should collect the sample or the separated acetone here. Okay, next one is fractional distillation. What is fractional distillation? When it is used, when the components in a mixture has the boiling point difference which is less than 25 degrees centigrade, then this method is used for the separation. Suppose if you have A, B, C components in a mixture, if A has 90 degrees boiling point and this is 100 degrees centigrade boiling point and this is like 115 degrees centigrade, so the boiling points are very nearer, right? The difference is less than 25 degrees. For such separation, fractional distillation is used. So this method is mainly used for the separation of petroleum products, gases from the air, etc. Let us see the setup. It is as usual as the distillation setup, but only difference is fractionating column. What is this fractionating column? So it is situated or equipped in between the mouth of this distillation flask and the thermometer, or you can say the condenser, not thermometer, condenser. In between the condenser and distillation flask, this fractionating column is fitted. For what? This is the original picture of fractionated column, two types. So it is packed with the glass beds, that's it. For what is the purpose? When it is fitted like this, when vapors coming to this column, fractionated column, it provides the glass beds provides the repetitive condensing or the cooling of the separated vapors. Okay, then it will collected in this conical flask. The setup is same. The process is when you heat the mixture, the vapors coming out of this and this fractionating column allows them to condense fastly or repetitive cooling, through the repetitive cooling. Then it will go into the condenser, then it will become liquid and collected to this flask. Okay, this is the original picture. So for example is, the main example is, it is used in industrial purpose for the separation of petroleum products from the crude oil. When crude oil and when it is heated, in crude oil we have different kinds of products like paraffin, naphthalene, diesel, fuel oil, and everything, lubricant oil. This is used for road, right? So the boiling points difference is very less. They are narrow boiling points between these products. That's why this method is used. The setup or the glassware which is shown in previous slide is used in laboratory purpose, but this is like when you go to the industries, and you'll see the biggest furnaces throwing this distillation setup. Okay.